Hello friends and welcome. Today we are going to talk about Rhodophyta. The main objectives of today's deliberation are: number 1, to find out general characteristics of Rhodophyta; second, to know about occurrence of Rhodophyta; third, to gain information about range of thallus organization in Rhodophyta; fourth, to find out morphology and cell structure of Rhodophyta; fifth, to give a detailed insight about reproduction in Rhodophyta; and finally to gain knowledge about the life cycle of Rhodophyta. Let me begin with a brief introduction about algae. Algae are chlorophyll-bearing, simple, thalloid, autotrophic, and largely aquatic, both freshwater and marine organisms. They occur in a variety of other habitats: moist stones, soils, and wood. Some of them also occur in association with fungi, like lichens, and animals, for example on sloth bear. The form and size of algae is highly variable. The size ranges from the microscopic unicellular forms like Chlamydomonas to colonial forms like volvox and to the filamentous forms like Eulothrix and Spirogyra. The algae reproduce by vegetative, asexual, and sexual methods. Vegetative reproduction is by fragmentation. Each fragment develops into a thallus. Asexual reproduction is by the production of different types of spores, the most common being the zoospores. They are flagellated, that's motile, and on germination give rise to new plants. Sexual reproduction takes place through fusion of two gametes. These gametes can be flagellated and similar in size as in Chlamydomonas, or non-flagellated, that is non-motile but similar in size as in Spirogyra. Such reproduction is called isogamous. Fusion of two gametes dissimilar in size as in some species of Chlamydomonas is termed as anisogamous. Fusion between one large non-motile, that is static female gamete, and a smaller motile male gamete is termed oogamous, for example volvox and fucus. The Rhodophyceae or red algae comprise the only class in the division Rhodophyta. The Rhodophyceae are probably one of the oldest groups of eukaryotic algae. The members of Rhodophyceae are commonly called red algae because of the predominance of the red pigment or phycoerythrin in their body. Majority of the red algae are marine, with greater concentrations found in the warmer areas. They occur in both well-lighted regions close to the surface of water and also at great depths in oceans where relatively little light penetrates. The red thalli of most of the red algae are multicellular. Some of them have complex body organization. The food is stored as Floridian starch, which is very similar to amylopectin and glycogen in structure. Most of the world's seaweeds belong to this group. The chloroplasts of red algae are believed to be derived from cyanobacteria that formed an ancient symbiotic relationship with the algae. The red algae mostly consist of seaweeds but also include the genera of free-living unicellular microalgae. The morphological characters of the class Bangiophyceae range from unicells to multicellular filaments or sheet-like thalli. The Rhodophyceae include morphologically complex red algae. Rhodophyta inhabit prevalently marine ecosystems, but they are also present in freshwater and terrestrial environment. Now let us see the occurrence of Rhodophyta. Red algae are mostly marine, although some freshwater forms, less than 100 species, are also found. They are most abundant in the tropics and occur at greater depths than other algae, up to about 268 meters. There is a marked shift in their abundance from the equator to colder seas. There are 10,000 described species of red algae worldwide, most marine, 3% freshwater. They have wide range of occurrence such as epiphytes on other algae, seagrasses or mangrove roots, epizoic on animals, epiphytic on pebbles and rocks, and samophilic in sand. The rhodophycean organisms range from autotrophic, independent complete plants to completely heterotrophic parasites. This include non-obligate epiphytes, Acrostium, rhodocotton complex, obligate epiphytes, Polysiphonia lanosa on Ascophyllum, semiparasites which have some photosynthetic pigments, and parasites with no photosynthetic machinery. Parasitic red algae are divided into adelphoparasites closely related to hosts and hence easier to establish pit connections, and alloparasites not closely related to their hosts. Now coming to general characteristics of Rhodophyta. Red algae have a number of characteristics that in combination distinguish them from other eukaryotic groups. Number 1, there is complete absence of flagella and centrioles. Second, Floridian starch as a storage product and storage of starch in the cytoplasm. Third, their photosynthetic pigments include phycoerythrin, phycocyanin, and allophycocyanin together with chlorophyll D and carotenoid teraxanthin. Fourth, there is presence of unstaggered thylakoids in the plastids. Fifth, there is lack of chloroplast endoplasmic reticulum. Sixth, polysulfate esters are characteristic components of the cell wall. Seventh, they exhibit a highly specialized process of sexual reproduction, that's oogamy, and vegetative reproduction is by fragmentation. Eighth, one of the striking features of most of the red algae is the presence of a pore-like opening pit connections in the cell wall through which cytoplasmic connection is maintained from cell to cell. Ninth, majority of the members exhibit triphasic life cycle. Now let us talk about the morphology of Rhodophyta. As in the green algae, thallus structures in the Rhodophyta range from simple to complex. Unicellular forms, of course, have the simplest structure. There are 5 unicellular red algae, but Porphyridium is a common unicellular species found in many freshwater pools. Higher in complexity than single cells or the simple filaments, branched or unbranched. Simple filaments are abundant among the red algae. In some cases, these filaments are alternate life history stages of an alga of more complex thallus form. The simplest filaments are uniseriate, forming a single line of connected cells. Multiple cells in each segment define a multiseriate filament, while even more complex forms are organized into corticated or polysiphonous thalli. Let's see the cell structure in Rhodophyta. The major feature of a red algal cell are a chloroplast with 1 thylakoid per band and no chloroplast endoplasmic reticulum. Floridian starch grains in the cytoplasm outside the chloroplast. No flagella, pit connections between cells in filamentous genera, and a eukaryotic type of nucleus. The detailed description of cell structure includes: Number 1, cell walls. Cellulose forms the microfibril framework in most rhodophycean cell walls. Although in the haploid phase of the Bangiales, that's Bangia and Porphyra, a beta-1,3 linked xylem, this polysaccharide composed of xylose residues performs this function. Unicellular red algae have an amorphous matrix of sulfated polysaccharides without cellulose surrounding the cells. The amorphous polysaccharides or mucilages occur between the cellulose microfibrils in the rest of the red algae. The 2 largest groups of amorphous mucilages are the agars and the carrageenans. These mucilages may constitute up to 70% of the dry weight of the cell wall. Cuticles, composed mostly of protein, can occur outside the cell wall. Number 2, Chloroplasts and storage product. Chloroplasts are usually stellate with a central pyronoid in the morphologically simple Rhodophyceae, whereas in the remainder of the Rhodophyceae they are commonly discoid. In the Rhodophyceae with apical growth, the chloroplasts usually originate from small colorless proplastids with few thylakoids in the apical cell. Chloroplasts are surrounded by the two membranes of chloroplast envelope with no chloroplast endoplasmic reticulum present. Thylakoids occur singly inside the chloroplast. The phycobilin pigments are localized into phycobilisomes on the surface of thylakoids, a situation similar to that in the cyanobacteria. Chlorophyll A is in the chloroplast. There have been erroneous reports of chlorophyll D occurring in the chloroplast. The phycobiliproteins include R-phycocyanin, L-phycocyanin, and three forms of phycoerythrin. The phycoerythrins being present in the greatest amount, giving the algae their pinkish color. B-phycoerythrin is present in the more primitive red algae. R-phycoerythrin occurs in most of the higher red algae, and C-phycoerythrin occurs in the Porphyridium, Porphyra, and Polysiphonia. Fluoridoside. O-alpha D-galactopyranosyl 1,2-glycerol is the major product of the photosynthesis in the red algae, although mannitol, sorbitol, digenoside, and dulcitol also occur. The concentration of fluoridoside increases in red algal cells as the salinity of the medium increases. Fluoridian starch is the long-term storage product occurring as grains in the cytoplasm outside of the chloroplast. Fluoridian starch is similar to the amylopectin of the higher plants, staining red-violet with iodine. In the more primitive Rhodophyceae, the starch grains are clustered as a sheath around the pyronoid of the chloroplast, whereas in the more advanced Rhodophyceae, the starch grains are scattered in the cytoplasm. Number 3, pit connections. Pit connections occur between the cells in all of the orders except the porphyridiales and the haploid phase of the bangiales. It has been pointed out that the term pit connection is inappropriate because the structure is neither a pit nor a connection. However, because the term has been used for so long, it is probably best to retain it. A pit connection consists of a proteinaceous plug core in between two thallus cells. Number 4, calcification. All members of the corallineales and some of the nymaliales Galaxora deposit calcium carbonate extracellularly in the cell walls. Anhydrous calcium carbonate occurs in two crystalline forms, calcite and aragonite. The two forms differ in specific gravity, hardness, and solubility. The corallineales deposit calcium carbonate primarily as calcite, whereas the calcified members of the nymaliales deposit calcium carbonate primarily as aragonite. In Ligora nymaliales, the aragonite occurs as needle-like crystals in the wall, whereas in the corallineales, the calcite occurs as massive deposits. Calcified walls of living cells probably have a mucilaginous component that slows the loss of calcium dipositive into the medium. If a calcified thallus is killed, the dispersal of the calcified wall is greatly accelerated. Number 5, secretory cells. Secretory cells or vesicular cells occur in some Rhodophyceae. These cells are colorless at maturity and commonly have a large central vacuole. The secretory cells in the bone mesonia are prominent and associated with high concentration of iodine. The concentration of iodine can be high enough to produce a blue color in herbarium paper with starch as filler, the chemical test for starch. Secretory cells are vestigial, lacking the large vacuole with its refractile contents, when these algae are grown in a medium without bromine, bromine can also occur as granular deposits in mucilage, such as in the thallus medulla of Thysanocladia densa or in the cuticle of Polysiphonia nigricans. Other types of secretory cells not associated with the accumulation of halogens occur. The cells are often called secretory cells even though they are apparently not involved in the secretion. In Antithamnion, these cells have a large centrivicule containing sulfated acidic polysaccharide. Now let us discuss range of thallus organization in Rhodophyta. The thallus structure in Rhodophyta ranges from relatively simple to multiaxial forms. Species of Bangiophysidae are unicellular, palmoid, simple or branched filaments and never show aggregation of filaments into pseudoparenchymatous structure as is characteristic of the Rhodophysidae. The members of Rhodophysidae reveal either uniaxial or multiaxial construction. In uniaxial forms, the main axis is composed of a row of usually large cells, while in the multiaxial forms, the main axis consists of more than one filament, arranged in a parallel manner. The range of vegetative structure in red algae can be described as: first, unicellular. It comprises of a single cell and is the simplest thallus. The unicellular plant body may be spherical, oblong, or pear-shaped, and sometimes elongated and approximately circular in cross-section. For example, Porphyridium. Second, palmoid. In this type, the individual cells have mucilaginous sheaths around them and are enveloped in a common gelatinous matrix of indefinite shape which may be microscopic or macroscopic in size. For example, Asterocystis. Third, filamentous. The simplest filamentous algae consists of a thallus of a single chain of cells. It results if the cells are held tightly by the middle lamella after multiple transverse divisions, that is, when a unicell divides several times in a single plane. When the filamentous cells undergo longitudinal divisions, the filament branches, for example, goniotrichum. Fourth, heterotrichous type. This is the most highly evolved type of plant body, showing a good amount of division of labor. Heterotrichy means presence of two types of branching: prostrate branching forming the prostrate system and erect branches forming the erect system. The prostrate system creeps along the substratum and sends rhizoids to fix the thallus. From the prostrate system arise the upright filaments forming the erect system, for example, Acrostium. Fifth, discoid prostrate type. In this type, the filaments of thallus are prostrate and give circular disc appearance, for example, Erythrocladia. Sixth, crusts or cushions. In this type, the prostrate system is reduced and the erect system is well developed. This gives the body a crust or cushion type of appearance, for example, Hildenbrandia. Seventh, elaborated erect system. The erect system develops from the prostrate system and is composed of one or more and usually branched photosynthetic filaments. In this type, complete absence of prostrate system is noticed and there is well-developed erect system, for example, Batrachospermum. Eighth, compact pseudoparenchymatous type, uniaxial. As indicated by the term pseudo, which means false, the plant body gives the appearance of parenchymatous construction. Pseudoparenchymatous algae have thalli that superficially resemble parenchyma, but which are actually composed of appressed filaments or amorphous cell aggregates. A thallus which possesses a single axial filament having branched laterals with or without cortication is called a uniaxial filament. The uniaxial construction in simple form showing clearly the filamentous nature. For example, Limenia. Ninth, compact pseudoparenchymatous type, multiaxial. Thalli, which possess a tuft of axial filaments with many laterals radiating out to the margins, are called multiaxial filaments. In this, one or many threads, uni or multi, of the erect system take part in the production of the mature thallus, for example, mammalion. 10th, foliose parenchymatous type. A membranous or foliaceous body results if the cells of a filament divide in two planes, that is, transversely and longitudinally, for example, porphyra. Now let us know about the reproduction in Rhodophyta. The reproduction takes place by means of vegetative, asexual and sexual methods. First, let us discuss vegetative reproduction. In majority of cases, it takes place by fragmentation. Second, asexual reproduction. Spores are non-motile and are given different names depending on the nature of cells from which they are produced and their number within each sporangium, namely monospores formed single in monosporangia, neutral spores formed by direct transformation of vegetative cells into spores, carpospores formed by division of zygote, and bispores, tetraspores and polyspores formed within diploid tetrasporangium in multiples of 2, 4 and multiples of 4. The asexual reproduction of the gametophytes takes place by neutral spores, monospores and polyspores. The neutral spores develop in ordinary cells of thallus, for example, Asterocystis. The monospores are developed in sporangia. A single spore develops in each sporangium, for example, Synenia. The polyspores are formed in large number in the sporangium. The asexual reproduction of the sporophytes takes place by tetraspores formed in tetrads in the tetrasporangia. For example, Polysiphonia and paraspores born inside parasporangium in greater numbers. During the development of tetraspores, reduction division takes place. In the development of the paraspores, there is no reduction division. Spores, whether monospores, tetraspores or corpospores, of almost all red algae are capable of motility by gliding. Some spores have smooth, directional and continuous gliding, for example, Batrachospermum. In others, movement is non-continuous and unidirectional. Polysaccharide secretion appears to be responsible for the gliding. Number 3, sexual reproduction. The sexual reproduction is always oogamous. The oogamy is of special type. The sex organs of Rhodophyceae have a distinguished terminology. The male structures are called spermatangia, developing non-motile spermatia, male gametes in them. The female sex organ is called the procarp. It has a carpogonium bearing a receptive structure, trichogyne. The egg develops in the basal swollen part of carpogonium. The post-fertilization events vary from one order to another. The more advanced orders have auxiliary cells with which the fertilized carpogonium fuses to form multinucleate fusion cell. Papin first recognized two types of auxiliary cells, nutritive and generative. Nutritive auxiliary cells provide nutrients for the developing carposporophyte, whereas generative auxiliary cells give rise to gonymoblast filament. The diploid tissue formed from the fertilized carpogonium forms the gonymoblast filaments. The gonymoblast filaments produce terminal carposporangia, which in turn form the carpospores. The carposporangia enlarge considerably during their maturation because of the development of the chloroplasts and the vesicles containing wall precursors. The pit connection between the carposporangium and the gonymoblast breaks before release of the carpospore. Also, during the development of the gonymoblast filaments, the pit connections between the older gonymoblast cells usually dissolve. Carpogonium. The female organ or corpogonium consists of a dilated basal portion and a usually narrow gelatinous elongate tip, the trichogyne, which receives the male cells. Usually there are two nuclei in a corpogonium, one in the trichogyne which degenerates soon after the corpogonium matures, and one in the basal part of the corpogonium which functions as the female gamete nucleus. In most Rhodophyceae, the corpogonium terminates a short, often branch 3 to 4 cell lateral called the corpogonial branch. The cell from which the corpogonial branch arises is the supporting cell. The corpogonium and the corpogonial branch are commonly colorless, although in some mammalians this is not true. Spermatium. The spermatia of the Rhodophyceae are spherical or oblong cells produced in spermatangia, a single spermatium being formed in a spermatangium and then released, leaving the empty sporangium. The spermatangia are formed or spermatangial mother cells. The young spermatangia frequently have a pronounced polar orientation, with the nucleus in the apical portion and one or more vacuoles in the basal portion. As the spermatangium ages, vacuoles form in the basal area. These vacuoles contain fibrous material, probably mucopolysaccharides, and make up half the volume of the spermatangium. Subsequently, the vacuoles fuse to form one large vacuole. The spermatium is released by the gelatinization of the spermatangial wall near the apex and the concurrent release of the fibrous material in the basal vacuole. The fibrous material presumably swells and pushes the spermatium out of the spermatangium. The fibrous material is sticky and some of it adheres to the spermatium, thereby facilitating attachment to the trichogyne. During the development of the spermatium, the pit connections with the spermatium mother cell is severed. The mature spermatium is uninucleate and wall-less, but surrounded by mucilage and may or may not contain functional chloroplasts. Fertilization. The spermatium usually is carried passively by water currents to the trichogyne of the corpogonium, although some spermatia can glide in a manner similar to the gliding in Porphyridium. Actin filaments in the spermatium and corpogonium are involved in the subsequent steps. The wall of the spermatium and that of the corpogonium dissolve. The male nucleus divides and the male nuclei move into the corpogonium. Fusion of one male nucleus and the corpogonial nucleus occurs in the basal portion of the corpogonium. Fertilization stimulates the production of polyamine spermine, which steers the corpogonial branch towards the production of carpospores. The trichogyne will usually continue to grow until contact is made with a spermatium. After fertilization has occurred, the trichogyne becomes separated at its base from the rest of the corpogonium by the progressive thickening of the cell wall. There is a relatively low statistical probability that the non-motile spermatia will be carried to receptive trichogynes of corpogonia. Some species have appendages on the spermatia that extend the reach of the spermatia 5 to 10-fold, resulting in a better chance of attaching to a receptive corpogonium. The appendages are initially contained within vesicles within the spermatangia and unfold when the spermatia are released. The appendage increases the surface area of the spermatium more than 30-fold, increasing the likelihood of interaction with a trichogyne of a corpogonium. Moieties of the sugar mannose cover the surface of the appendages. The surface of the trichogyne is covered with the lectin concanavalin A, which binds to the mannose moieties on the spermatangial appendages. Now coming to the zygote and its germination. In subclass Bangioideae, the zygote divides by vertical and transverse divisions. The number of cells may be 2, 4, and 16. The spores are known as carpospores. They are naked and liberated by disintegration of zygote wall and move about in amoeboid fashion. The first division of the nucleus of the zygote is reductional. In subclass Floridae, the carpospores develop indirectly from the zygote. Finally, let us talk about the life cycle of Rhodophyta. In the life cycle stages of Rhodophyceae, three phases are quite distinct, that is a gametophyte, a carposporophyte, and a tetrasporophyte. Thus, the life cycle is trigenic or triphasic. They display alteration of generations. In addition to gametophyte generation, many have two sporophyte generations, the carposporophyte producing carpospores and tetrasporophyte, this produces spore tetrads, which dissociate and germinate into gametophytes. The gametophyte is typically but not always identical to the tetrasporophyte. Carpospores may also germinate directly into thalloid gametophytes, or the carposporophytes may produce a tetraspore without going through a free-living tetrasporophyte phase. Tetrasporangia may be arranged in a row, that is zonate, in a cross, cruciate, or in a tetrad. The carposporophyte may be enclosed within the gametophyte, which may cover it with branches to form a cystocarp. In the carposporophyte, a spermatium merges with a trichogyne, a long hair on the female sexual organ, which then divides to form carposporangia, which produce carpospores. Carpospores germinate into gametophytes, which produce sporophytes. Both of these are very similar. They produce monospores from monosporangia just below a cross wall in a filament, and their spores are liberated through apex of sporangial cell. The spores of a sporophyte produce either tetrasporophytes. Monospores produced by this phase germinate immediately with no resting phase to form an identical copy of parent. Tetrasporophytes may also produce a carpospore, which germinates to form another tetrasporophyte. With this, we conclude today's lecture. Hope this was helpful. Thank you.